as Brian mentioned, I will talk about the systemic therapy for advanced melanoma. Before I start, I'd like to acknowledge people who are live streaming into this meeting, and I would like to thank them for doing this and remind everybody that they can actually send emails with the questions into the meeting that we will answer at the end of the meeting. Also, for those here, please feel free to write your questions down, and we'll take all of these, collect all of these during the break and at the end, and take all of your questions. So my talk is about systemic therapy for advanced melanoma. Basically, these are stages of melanoma that require medications, drugs, to treat the disease. And these are my disclosures. I've done academic and scientific consulting for these companies. I've received recent research grant support from these companies as well. So the main focus of my talk is about stage 4 melanoma, meaning, as Brian mentioned, these are patients whose tumor has spread beyond the primary site of the tumor into other organs, such as the brain, the lungs, the liver, and so on. So when we talk about the medications that were available for treating this disease and were approved by the FDA, before 1998, most of the approvals were made based on small studies that have only shown that these drugs are safe and possibly have some activity. And in that era, we had chemotherapy, a drug called dacarbazine. We had immunotherapy in 1998, a drug called interleukin-2 or high-dose IL-2. And at the time, the survival, the expected survival at 1 year was less than 25%. Between 1998 and 2011, we had absolutely no approvals from the FDA to treat this disease. However, there was a lot of research happening in the labs, in the clinic, and then suddenly we had an explosion, basically a revolution starting 2011, basically 10 approvals, 10 different approvals between 2011 and 2015 for treating stage 4 melanoma. So where we stand today, the 1-year survival is more than 70%, major progress since then. So we can divide the agents available for treating melanoma into 3 main categories. What we call immunotherapy, that basically activates the patient's own immune system to fight the cancer. What we call targeted therapy, these are medications that are designed to specifically target the cancer. And chemotherapy. Starting with chemotherapy, basically this is something that we used to use in the past. As I said, dacarbazine was approved in 1975. It has some activity. We rarely use it today to treat this disease, but it's still there, on the shelf if needed. Targeted therapy, we have 4 medications listed here: vemurafenib, dabrafenib, trametinib, and cobimetinib. And these were actually, these kind of highlight a revolution in treating melanoma based on a significant discovery that was published in a major journal in 2002. It was published at the time that about 40 to 50% of patients with melanoma, their tumors have a specific mutation, a defect in the DNA. It's called BRAF. What does this BRAF do? Let's look at this graphic, and this brown circle is, think of it as the cancer cell. These cancer cells are driven to survive and divide and make more cancer cells. And the way they do this, there are certain signals that turn on specific pathways within the cancer cell, ordering it to divide or stop dividing or grow or stop growing. In a normal cell, you have a signal that activates the pathway on and off, and that's how our normal cells work. In a cancer cell like melanoma, where we have this mutation or defect in the BRAF gene, it results in a so-called BRAF protein, which acts like an engine which is firing all the time, that basically does not shut off. And it's independent of any signaling above. So basically, it's just out of control. And this gave the rationale to do research and design drugs that can actually block this protein, this engine, shut it off, and therefore kill the cancer cell. And that's how multiple drugs were tested and have shown significant activity. And this led to this major advance, multiple publications, series of publications, some of the fastest advances in research, leading to what we have today in the clinic, a drug called vemurafenib, another drug called dabrafenib. When we look at the major studies that tested these drugs, if you look at these graphs, so above the graph, these are the cancer continue to grow. Below the line here, means that the cancer has shrunk. So as you can see, when we give these drugs, the majority of these cancers actually shrink. And about 50% of them have what we call an objective response, meaning at least 30% reduction in the size of the cancer. And this 50% response rate, and these were relatively what we call durable responses, led to the approval of these medications by the FDA. However, there was a problem. Although we have at least 50% objective major responses, about 50% of patients progressed in about 7 months from starting this disease, meaning there were other resistance mechanisms, things that making these drugs not to work anymore. And we look at these, there was a lot of research was done in the labs all over the world, looking into those resistance mechanisms. And here again, this is the BRAF protein. So we're trying to block this BRAF protein with the medication. The tumor cell, what it does is it learns to evade this blockade and activate a downstream protein. So now we have more proteins inside the cancer cell activating the protein below. It's called MEK. So now we're blocking BRAF, but we have activation through MEK from different ways, different pathways. And this gave the rationale that maybe we should actually block both proteins, so BRAF and MEK. And this was shown to actually work much better in animal models, in mouse models, and then translated into human studies. And major studies, such as these two studies here, have shown that now when we combine these two medications, basically blocking both BRAF and MEK, so here with dabrafenib plus trametinib or vemurafenib plus cobimetinib, the response rate now improves from about 50% with one drug to about 60 to 70% with both drugs. Not only that, on average, these responses are more durable. They last at least 9 to 11 months. So 50% of patients, the response will last more than 9 to 11 months. And this led to the approval of these combinations, and that's what we use today in clinic for patients whose tumor has the BRAF mutation. Moving on to immune therapy, as I said, these are medications that activate the immune system to fight the cancer. And this is the list of medications approved in the clinic. We have interleukin-2, ipilimumab, pembrolizumab, nivolumab, and T-VEC. So thinking how we activate the immune system, think about this brown circle here as the cancer cell again. And these blue circles as the, let's say, the soldiers that are sent by the immune system to fight the cancer cell. We call them T cells. There are two different ways of making T cells to go fight the cancer. One of them is called passive immunotherapy. We do surgery, take the tumor, and take these T cells, grow them in the lab, and give them back to the patient. Another way is called active immunotherapy. Basically, give drugs that activate, stimulate the immune system to make more T cells that will go and fight the cancer. And we have a lot of medications, different ways of making more T cells. We have so-called cytokines, like interleukin-2 or interferon alpha. We have cancer vaccines, basically give a vaccine from the cancer so that the immune system will make more T cells to fight the cancer. And when these T cells make it recent research has discovered that there are certain, let's say, checkpoints or certain agents on the T cells that can be manipulated to turn them on or off. So interleukin-2 is the drug approved in 1998. This drug was a cytokine, basically a blind immunotherapy, which is given. And what we learned is that about 6% of patients will achieve what we call a complete response. Basically, that the cancer disappears. More importantly, about 60% of those complete responders, meaning about 4% of the total population treated, actually had durable responses beyond 10 years. And this was the first clue that we can actually cure melanoma with immunotherapy. And this was approved in 1998. It was a small percentage of patients, but these patients were cured. And this kind of sparked this more interest in investigating how we can make the immune system fight the cancer and hopefully cure more patients. And so this kind of led to this new exciting area of so-called checkpoint inhibitors. Here again, think of this brown circle as the cancer cell, and this blue circle as the T cell, the soldier made by the immune system to fight the cancer. So the way it works, we have a control center in the immune system, so-called dendritic cell, that basically programs the T cell to go fight the cancer. If we zoom in at this interaction between the control center and the T cell, there are certain interactions that lead to the activation of T cell to send it go fight the cancer. But the immune system also has checks and balances. When we have overactivation of the immune system, we know that we can develop autoimmunity, diseases like rheumatoid arthritis, lupus, etc. So the immune system has brakes that can basically put control over the activation of these T cells. One of those brakes is called CTLA4. So this is important physiologically to avoid autoimmune diseases. But if this T cell is a T cell that is meant to fight the cancer, CTLA4 is doing the wrong job here. It's preventing the T cell from doing this job. And this gave the rationale for designing drugs that can block CTLA4, a drug called ipilimumab, and thus allow this T cell to do its job and basically migrate into the area of the cancer and fight the cancer. When the T cell makes it to the tumor cell area, to the cancer cell area, and here again, if we zoom in, we see there are certain interactions between the T cell and the cancer cell. The T cell is trying to kill the cancer cell, but the cancer cell has more tricks up its sleeve. It can actually inhibit the T cell by basically linking into another inhibitory signal called PD-1. It acts like CTLA4, basically dampens or basically breaks the T cell from doing its job. And this gave the rationale to develop drugs to block this interaction, so-called nivolumab and pembrolizumab, which can also basically salvage or save this T cell from inhibition from the tumor and allow the T cell to go fight the cancer and kill the cancer cell. So what happened in the clinic? Starting with this drug ipilimumab, the CTLA4 inhibitor, studies were done. This drug works well. About 15 to 20% of patients respond. More importantly, if we look at the proportion of patients alive 5, 9, 10 years later, we see that the survival curve, which was about 4% with IL-2, with interleukin-2, now it jumped up to 21%. It was a major thing in melanoma. This was back in 2011. It was kind of a major step in hopefully curing this disease. Now, as I said, this is a physiologic mechanism to break the immune system. So what you expect is that the side effects are what we call autoimmunity. The immune system will be hyperactive and cause some other side effects that Changmin Song, our nurse practitioner, will describe and discuss after the break. Now, moving on to the other drugs which work at the level of the tumor, at the tumor level, the PD-1 inhibitors, nivolumab and pembrolizumab. These were also tested in major studies, and in actually more than two major studies have shown that these drugs also work very well. These can shrink the tumor in about 40 to 40% to 45% of patients. Essentially, they can decrease the risk of death from melanoma by about 58% versus chemotherapy, and by about 30 to 37% versus the drug ipilimumab, the CTLA4 inhibitor. And this obviously led to the regulatory approval of these drugs to treat this disease. Someone may tell me, well, okay, we tested ipilimumab at this level here, it works. We tested these drugs here, PD-1 inhibitors, they work. Why not combine them? Basically, make more T cells, send them to the tumor environment, and then salvage them, making them work better in that area. And these studies were done. In fact, we actually did a study where we kind of supported this idea of combining the drugs. Our patients volunteered to give us a biopsy before we give any drug. Then we give them the drug ipilimumab, the CTLA4 inhibitor. Then we did another biopsy 6 weeks later. And we saw that there is a massive infiltration of these T cells, these brown dots, into the tumor microenvironment. And so basically we're making more T cells, we're sending them to the tumor microenvironment. Now, why not add the PD-1 inhibitor and make them work better? So a major study was done. It's a so-called phase 3 study, international study, which basically combined CTLA4 with PD-1 versus PD-1 alone versus the drug ipilimumab. So this is nivolumab plus ipilimumab. This is nivolumab alone. This is ipilimumab alone. And what we saw is that basically, as you might expect, the likelihood of shrinking the tumor was better with a combination. It was about 59% of patients will have their tumor shrink by 30% or more. About 45% of patients will have their tumor shrink with monotherapy, with one drug alone, the nivolumab, and with the older drug, ipilimumab, about 19%. Importantly, all these responses were durable. In fact, the majority of patients who respond to these immunotherapy drugs end up having long-term durable responses that go on for many months and probably years. However, this improvement in response comes at a cost, which is toxicity. We said that these checkpoints, they put brakes in the immune system. They make us not get autoimmunity, not get lupus, rheumatoid, etc. So when we block more of them, we'll get more side effects. And this is what we see. So what we call here grade 3-4, these are so-called serious adverse events. So when we combine nivolumab with ipilimumab, the likelihood of getting serious adverse events was about 59%. When we give PD-1 inhibitor alone, nivolumab, was about 21%. When we give the old drug ipilimumab, was about 28%. Importantly also, when we look at long-term survival, we're not sure at this time, even if we get a higher response rate, that we're actually having a significant improvement in survival by using the combination or monotherapy. So we're selective in using these combinations, taking into account toxicity, patient preference, the need for a response, and the available agents and so on. So we use them, of course, but we're selective, and we kind of tailor this to the patient and needs. An exciting drug also, which is now available in the clinic, is actually a genetically engineered herpes virus. This is the herpes virus, which is genetically engineered to basically invade and kill the cancer cells. So this is injected into the cancer cell as an injection under the skin. It leads to destruction of the tumor cells, but also helps attract the immune system cells to come to the tumor cell area and basically help the immune system also kind of identify and fight the cancer cell. Studies have shown that this can cause about a response rate, so-called durable response lasting at least 6 months, of about 16%, leading to regulatory approval of this drug as well. So basically, bottom line, the key is, as I said, is survival, right? Are we making progress on the long term in the survival of our patients? And we can look at these survival curves from chemotherapy to target therapy to immunotherapy and so on. And you can see that these survival curves continue to go up and up and up. So today we expect that at least 50 to 60% of patients will be alive 3 years and beyond. And this is the latest data, so hopefully we will have 5-year data, 10-year data soon. But we expect at least 50 to 60% of those with stage 4 will be alive beyond 3 years. So where we're going in the future, it's combinations, smart combinations. How can we combine drugs to make more T cells? Or how can we combine target therapy with immunotherapy? Ideally, make these responses 70, 80, 90% and make them durable responses. And that's the ongoing research. And we're there already. So this is a study we're conducting at the Eastern Cooperative Oncology Group. We're in this era of triple combinations. So this is only one example of triple combination studies, which is ongoing. Another question that we always get is somebody has a BRAF mutation. Should we do immunotherapy or target therapy? So this is another national study where patients start with target therapy. Another group starts with immunotherapy, and we follow them. As long as they're doing okay, they continue. If they progress, they cross over to the other drug, and they continue from there. And this will give us information on the long term. Should we start these patients with immunotherapy or targeted therapy? So these recent advances in the immunotherapy has actually moved us now into this new era of long-term survival and, yes, achievable cures. And we believe we can cure this disease for some patients. So briefly, I will talk about adjuvant therapy of melanoma. And these are patients with stage 3 that Dr. Gassman mentioned. We do surgery for them, but the cancer has already made it into the lymph nodes. These are patients considered at high risk that the cancer may come back. So we give them immune therapy to hopefully cure this disease before it progresses into stage 4, before it comes back. And I know, some of you are belong to this group here and getting these drugs. So what are the drugs approved by the FDA? How well they work? We had, until recently, 3 drugs. One so-called interferon alpha, pegylated interferon, ipilimumab at 10 milligram per kilogram. This worked modestly, had toxicity, and luckily, only recently in the fall of 2017, we had 2 major studies that have shown that this PD-1 inhibitor, nivolumab, actually works well, and it is less toxic. So this is a new standard of care. For patients with BRAF mutation in their tumor, the combination of BRAF and MEK inhibitors, the dabrafenib and trametinib, also works well for this group of patients, stage 3. It's now FDA approved as well. And most recently, only a couple of weeks ago, the results of this study were reported testing the drug pembrolizumab. This is the other immunotherapy PD-1 inhibitor that we expect will achieve regulatory approval. Now, another final question is the question of personalized therapy. You come to clinic, we give you the same drug, everybody. But why some patients respond and others don't? Can we do some testing that can tell us what patients are more likely to benefit from this drug, but not that drug? For example, in immunotherapy, what patients are more likely to get toxicity or not? This is an important area of research, which is kind of expected to revolutionize basically and change the way we treat patients, basically make it personalized medicine for you only. And some of the examples is a report that we published recently where we looked at the genes in the tumor and looked what genes are turned on, what genes are turned off. And based on this profile of these genes in the cancer cell, we were able to divide patients into those who are most likely to benefit from immunotherapy versus those who are least likely to benefit from immunotherapy versus those that are in the middle. Another report we recently published as well is looking at those who may get the toxicity of colitis or diarrhea from immunotherapy. And we found that a marker in the blood called interleukin 17, when it's high, it predicts patients that are more likely to get this toxicity. So these are considered preliminary data that are currently being validated in a national study. But this is where we're heading, and this is where this disease is going. So thank you very much for your attention. We'll